let's go back to page 81A. What I'd like to do, before I start looking at all this and you start writing down, you say, wait, is that, I haven't written it down. Let's look at a picture. Let's look at page 81C. Let's look at the bottom of 81C. All right, so this is a picture at the bottom of 81C of the eye. And the black spot in the center is the pupil. The pupil is really a hole. It's really a hole through which the rays of light go through. It's an opening, a hole. Surrounding that hole is a pigmented muscle called the iris. The iris is a muscle. It's a muscle that can contract or relax, and as it contracts or relaxes, it controls how wide open that hole is. And it's pigmented, so the muscle may look blue or green or brown or hazel or whatever. Just next to, outside that pigmented muscle, is an area we call the whites of the eye, which is really the eyeball. It's really the eyeball. And the eyeball is hard, and it's called the sclera. The sclera. Sclera means hard. All right, now, let's look at the top picture. This is a cutaway side view of the eyeball. So, this whole outer part of the eyeball, this outer part is called the sclera. Sclera means hard. Now, on the wall of the eyeball is made up of 3 layers. The outermost layer is that sclera, just hard. It's fibrous connective tissue. It's real tough. All right, right underneath the sclera is an area called the choroid. The choroid. Now, the word choroid reminds us of the word choroid plexus. We had learned that in the brain, there's this vascularized membrane called the choroid plexus, and it forms cerebrospinal fluid in the brain. There is a vascularized membrane right underneath the sclera called the choroid. It's got a lot of blood vessels. You'd say, is it forming cerebrospinal fluid? No, it's not. But it is a vascularized membrane that provides a blood supply. Now, the innermost layer, which I colored red, it's not really red, the innermost layer is called the retina. The retina. The retina is where your photoreceptors are. That's where the photoreceptors are. The rays of light go through this clear membrane on the front of your eyeball called the cornea. When you rub the front of your eyeball, you're rubbing the cornea. All right? The rays of light then go through this chamber known as the aqueous humor, and then they pass through an opening called the pupil through the lens, and the lens focuses the rays of light onto the back of the eyeball, onto the retina, and where those rays of light converge, where they focus, is an area called the fovea, or fovea centralis. Now, we learned that the area right here, which is kind of around that fovea area, is where most of the cone photoreceptors are located. The cone photoreceptors allow us to see in color vision, and they also provide a high degree of resolution. You'd say, what's resolution? Sharpness. The cone photoreceptors are really what give us our sharpest vision. So they are concentrated in this area called the macula lutea. So that's where most of the cones are. Obviously, the retina also extends up here and down here. But that's where the rod photoreceptors are, which allow us to see in black and white and shades of gray in dim light. All right, so another thing we talked about last class meeting, back on page 81A. So on 81A, at the very bottom of 81A, so we talked about how there are the rod and cone photoreceptors, and we gave you a little bit of a sense of how they work. In these photoreceptors, there's a chemical. Now, in the rod photoreceptors, the name of the chemical is rhodopsin. In the cone photoreceptors, the name of the chemical is something else. They're known collectively as iodopsins. And this rhodopsin chemical, or whatever chemical is in the photoreceptor, is activated by light. And when light causes a chemical reaction, that's called a photochemical reaction, which is what we wrote here. We learned last time that rhodopsin is actually a protein combined with vitamin A, retinol. So that explains why you need vitamin A or retinol in order to see. And when we were learning the various vitamins at the end of section A, we mentioned that vitamin A is needed for this chemical reaction for vision. And in fact, we learned back in section A that a deficiency of vitamin A results in a condition known as night blindness. Why is it called night blindness and not blindness in general? Because the rod photoreceptors are primarily used to allow us to see at night in dim light. So the cone photoreceptors use a different photochemical. Anyhow, when light activates the rhodopsin, it leads to a photochemical reaction resulting in depolarization, and the depolarization in the neuron in this photoreceptor leads to an action potential. And now the action potential is on its way to your brain. But we learned at the very end of class last time on page 81D, 81D. So what are we looking at? Right here, it shows the eye. And we're looking, this boxed area is what we're looking at. So we're actually seeing the retina. And so the retina is more than just photoreceptors. It's the photoreceptors plus a whole bunch of other neurons. So the retina of the eye is actually quite complex. It's not just simple sensory neurons called the photoreceptors. These photoreceptors, and the tall ones are called the rods, and the cone-shaped ones are the cones. You'll notice they synapse onto other neurons. These neurons here are called bipolar cell neurons. We actually learned that term way back on page 35. You might remember back on page 35, we talked about how some neurons are multipolar shaped, some neurons are unipolar shaped, and some neurons are bipolar shaped. And the bipolar shaped neurons are found in only one place in our body, and that's in the retina of the eye. Now, I don't care that you know bipolar neurons. I'm just mentioning that we have talked about them before. The bipolar neurons then synapse onto what are called amacrine neuron cells, and the amacrine neuron cells synapse onto what are called ganglion neuron cells. I'm not asking you to know the names of any of these. I just want you to understand that the retina of the eye is more complicated than just sensory neurons. It's more complicated than just photoreceptors. There's already a lot of neural processing going on in the retina of your eye, even before the signals are sent to your brain. In other words, the signals, the information that is being sent from your eye to your brain is not just simple color information, black and white or color information. It's actually sending information about the shape of the objects you're looking at, and it's also sending information about movement. So there's a lot of neural processing occurring in the retina. Now, we've explained that the rod photoreceptors are activated, they contain rhodopsin, and they're activated, they're very sensitive to light, so they're activated in dim light. The cone photoreceptors come in three types because they have three different chemicals in these three types of cone photoreceptors. Now, I've colored them blue, green, and red. I did that to represent the fact that there is one group of cone photoreceptors in the retina of your eye that are activated by blue light. There's another set of cone photoreceptors in your retina of your eye that are activated by green light. And there are a third set of cone photoreceptors in the retina of your eye that are activated by red light. The chemicals that are inside them are called iodopsins, and there's three types of iodopsins. Now, if these three cone photoreceptors allow us to see in color, one obvious question is, how are we able to see all the different colors in the rainbow if there's only three cones? You know, one activated by blue, one activated by green, and one activated by red light. I mean, what about orange? What about purple? What about chartreuse? Whatever that is. So how are we able to see all these other colors? So let me give you a couple of analogies of how this works. Many of you have laser jet color printers. Now, I don't know how many inks you have. Maybe you've got one of the newer, fancier ones that has, what, 5 different inks? 7 different inks? But do you have an ink in your laser jet printer for every single color in the rainbow? So how are you able to print a picture with all the colors? Because it's a matter of blending. So by blending all these different colors, we can create all the different colors. And in fact, we don't even need to have 7 different colors. We don't need to have 5 different colors. Obviously, it'll work with just 3 different colors. A second example is on a color screen, right? This is a color screen right here. A color TV set, a color monitor, all right? So it has, what creates the color image on a TV screen, a monitor, are little pixels lighting up. Does anybody know what I'm talking about? These little round pixels light up. There's actually only 3 types of pixels. You know what those 3 colors of pixels are? Blue, green, and red. And by lighting up to different intensities, it creates all the colors that you see on a color TV set. So when somebody is looking at something that's orange, it activates these 3 types of cone photoreceptors to different degrees. And our brain is going to interpret the way they're activated as, quote, orange color, or purple color. So it is by a blending of these that we're able to see them. Now, I want to mention the subject of color blindness. Color blindness is a genetic disorder. It's genetic. And there are people who are born because of a genetic defect where they do not have all 3 types of cone photoreceptors. They might have only 2 of the 3. They might even have only 1 of the 3. They might, in fact, have none of the 3. So there are different types of color blindness. The most common type of color blindness is where the person is either missing either the green or the red cone photoreceptors. Because they're missing either the green or the red, they have trouble telling the difference between green and red. Now, green and red's kind of important to be able to tell the difference. Like when you're driving a car and there's signal lights. So let me ask you a question. Do they allow somebody who's colorblind to still drive a car? Yes, they do. Well, then how can they drive a car if they can't tell the order? That's right. They can't tell whether the light looks green or red, but if the top light is lit up, and they can tell that it's lit up, they just can't tell what color, they stop. Because, you maybe never thought about this, the red is always the top light. They go by position. They cannot go by color. They can't tell the difference. Now, when the question that always arises is people will say, well, if they can't tell red from green, then what does it look like to them? So to help you understand that better, let me give you a sense of this. All right, I'm going to show you how this works. All right, they have these tests for people to see if they can see in color. All right, now, can you see this? Do I need to make it bigger? I think you can make it out. Now, all of us, is there anybody who's colorblind in this class? No? Okay. So, all of us can see there is the number 8 here. But for somebody who can't tell green from red, they can't see that number because this looks exactly the same as that. Does everybody follow that? So that's because here's another example. All right? All of us can see the number 16. But somebody who can't tell red from blue can't see that. I'll give you a couple of other examples. Here's another one. What number do you see? 2. But somebody who can't tell green from red cannot see that number 2. So that's what we mean. That this color here looks absolutely the same as that color there to them. So they just can't tell the difference in color. So there's all these different color blindness tests that are given because there's different types of color blindness. Now, I mentioned I mentioned that color blindness is a genetic defect where the person doesn't have all three types of cone photoreceptors in their eye. Interestingly, this gene for making cone photoreceptors is carried on the X sex chromosome. When genes are carried on the sex chromosomes, they're said to be sex-linked. Now, this explains why most people who have color blindness are males. It is extremely rare in females to be colorblind, but it's not uncommon for males. Now, let's remember this. We learned this way back in section B. Males are XY, females are XX, right? So, so we know that females are XX. One of these X's they got from their mother, the other X they got from their father. Now, the only way that a female would be colorblind is if she has inherited a genetic defect for color vision on both X sex chromosomes. So if she has inherited a defect for colorblindness both from the X from her mother and the X from her father, then she'd be colorblind. That's not likely. Especially because it's especially not likely that she'd get it from her mother because women just don't have the color blindness. However, guys are XY. They only have one X sex chromosome. The Y does not provide any information about the ability to see in color. So the Y comes from which parent? The father. Only males have Ys. So the Y came from the father, so the X of a male who was XY came from his mother. If he inherits a defective gene from his mother, he's colorblind. Remember, a female would only be colorblind if she's inherited a defect from both her parents. So the way this is usually transmitted is the mother can see in color, but she's a carrier of this defect. One of her XX chromosomes, she's like this. That's the mother. And she transmits, there's a 50-50 chance that her son will inherit this X with the defect in color vision versus the normal one. And so if the mother is a carrier of the colorblind gene, there's a 50-50 chance that the son will inherit it. And he'll be colorblind. Because if he just has even the one defect. All right, does everybody follow that? That's the color blindness. One more thing about color vision. We as humans are really unique. We're one of the few species on the planet that sees in color. There's a few birds that see in color, a couple of tropical fish that see in color. Your dog does not see in color. Your cat does not see in color. Most animals do not see in color. Now, an obvious question that students always ask is, well, like, how do we know that? We can't get inside their mind. No, we can't get inside their mind, but they don't have cone photoreceptors in their eye. So they only have rod photoreceptors. So since they don't have cone photoreceptors, the assumption is they don't see in color. So dogs and cats just see in black and white and shades of gray. Now you can see fine that way. We've all seen some movie at some time or another in black and white, right? You can still watch the classic movie Casablanca, you know, with Humphrey Bogart and who's the woman, Ingrid Bergman, is that her name? I mean, however it is. So it's still a great movie, even if it's black and white. But you know, the dogs would, everything, all the movies dogs would watch would be in black and white. All right, so let's go back to page 81B. Actually, just before we do 81B, sorry. Let's look on page 81E. 81E. So on page 81E at the top, this is showing how these signals go from your eyeball through the optic nerve to the back of the brain. We learned last class meeting that the primary visual area is here at the back of the occipital lobe. So it's interesting that your eyes are in the front of your head, but where you actually convert those electrical impulses to a visual image is at the back of your brain. Now, here it shows that there's this cable called the optic nerve, cranial nerve number 2. It's got millions of wires sending signals about what your eye is seeing. And notice it goes to our old friend, the thalamus. Most sensory information is going to go to the thalamus, and then it will be relayed or forwarded to the correct area of the brain. Now remember, most sensory information, when it goes to the thalamus, most sensory information then is forwarded up to the primary sensory area at the top of the parietal lobe. But this is visual information, so that goes to the back of the brain, the occipital lobe. Now, in fact, this neural pathway is a little bit more complicated. You had to know that. It was going to be more complicated. So let's look at the bottom picture. This actually gives us more information. Okay. So what we're looking at is we're looking at a view of the brain. We're standing on top looking down, right? So here's the eyeballs, the right and left eyes. Here's the optic nerves, Now, haven't we learned that most sensory information decussates? Remember how usually the signals, the nerve fibers cross over or decussate, so that sensory information from the right hand ends up going to the left side of your brain? Remember we've learned that? Well, the vision is not any different, although there's one slight twist. Right here, here's the optic nerve with all these wires coming from the eye, and here's the area where they decussate. It's called the optic chiasm, which means to cross. Chiasm. Now, what is a little bit different is that most sensory information, essentially 100% of the nerve fibers decussate. In vision, only 50% of the nerve fibers decussate. So 50% of the wires from the left eye decussate over to the right side. 50% of the wires, the nerve fibers from the right eye, decussate to the left. But it's not 100%. Now, once these fibers cross, now we call these cables, we no longer call them optic nerves, we call them optic tracts. You'd say, yeah, like why? Because in the optic tract, half of the wires came from the left eye and half from the right eye. Half of the wires came from the left and half from the right. The optic nerve, all of the wires were coming from either the left or the right eye. So now they're mixed. Now, you'll notice here's the thalamus, and here in the optic tract, 75% of the wires in the optic tract synapse in the thalamus, 75%. And then from there, they travel in what's called the optic radiation to the primary visual area at the back of your brain. So an obvious question is, well, wait a second, why did you say only 75% of the wires in the optic tract synapse in the thalamus? The answer is because 25% of the wires in the optic tract synapsed right here. You'd say, what's that? The oculomotor reflex center in the midbrain. You'd say, what's the oculomotor reflex center? That's the control center that controls whether your pupils are dilate or constrict. So 25% of the wires that are in the optic tract that came from your eyes inform this control center, this homeostatic reflex center. They inform that center about what you're seeing and whether you need to increase the amount of light you're taking in or decrease the amount of light. The oculomotor reflex center is what's controlling how wide open you dilate your pupils or constrict them. And it has to get information, just a little bit of information about what you're seeing in order to know whether to dilate or constrict your pupils. It's also involved in focusing. We'll get into that later. All right, let me summarize what we've just covered here. Let's go back to page 81B. 81B. And I think you'll see that everything we've just shown you is summarized right here on page 81B. Nerve fibers are in the optic nerve, cranial nerve number 2. Decussation of half the nerve fibers occurs at the optic chiasm. The nerve fibers then travel in the optic tracts. About 25% of the nerve fibers synapse in the oculomotor reflex center, which is located in the midbrain and controls or regulates pupil size and accommodation of the eyes. You'd say, what's accommodation? Focusing. The remaining 75% of the nerve fibers, the majority of them, synapse in our old friend the thalamus. They travel through the optic radiation to the primary visual area, which is located in the occipital lobe of the cerebral cortex. So I think everything that we saw in that picture, I pretty much have written or summarized right here. I want to show you one last thing about the eye before we leave it. Take a look at page 81F. 81F looks very much like the picture that we saw on 81E at the bottom. Looks pretty much like the picture we saw on 81E at the bottom. But allows us to understand one more thing. Remember how we learned about the wiring of our spinal cord, and then we could ask questions of what would happen if there was a lesion or injury in the spinal nerve or the ventral root, or a lesion in the spinothalamic tract, or an injury to the corticospinal tract, and we could ask you what would be the symptoms if somebody had these different types of injuries. Well, we could do the same thing with the eye. Let's say that somebody severed or injured this optic nerve from their left eye. Now, in the optic nerve are wires just from the left eye. So that would mean they wouldn't be able to see anything in their left eye. Does that make sense? Look, there's a cable coming off your eyeball. If you cut that cable, you're not going to see anything that your left eye sees. The right eye is still sending information. All right? So it shows you in the bottom, right here, it shows you that damage in the left optic nerve, you lose vision in your left eye. From your left eye. You can still see with your right eye. But notice what happens here. Let's say, what if there was a lesion where it has number 3 in the optic tract? Now remember, in the optic tract, 50% of the wires came from the left eye, 50% of the wires are from the right eye in the optic tract. So if the optic tract is cut, you know what it looks like? You can still see half the visual field in each eye, and you've lost half the visual field in each eye. Does that make sense? Because 50% of the wires were from the left eye and 50% from the right. And so you've lost half from both eyes. So I would like you to understand the difference in what happens whether somebody injures their left, whether they injure their optic nerve or their optic tract.